All right, Ninja Nerds, we're going to talk about the layers of our blood vessels, or our tunic layers. So what I have over here, I pre-drew two distinct blood vessels. So I have over here on the left, I have an artery, and I have over there on the right, I have a vein. And what I want to do is, now their layering is pretty much going to have all the same things. It's just going to be about which one's thicker, all right, which one's going to be a little bit thinner. And then we might talk about just a couple abnormalities within this. And then I want to go over here and talk about capillaries, the three different types of capillaries. All right, so first things first. If we look here, this inner lining of the blood vessel, the tunica interna or the tunica intima, it actually, there is two distinct layers of it. So if you look here, there's an endothelial lining. So it's these little black cells here. These little black cells are simple squamous epithelial cells. And then right underneath it, this little blue lines right there, that's called the subendothelial layer. So it prevents the blowout, right, of the blood vessel. It's made up of collagen and some other types of proteoglycans and proteins. So it's kind of like a loose areolar connective tissue. So this right there is called our subendothelial layer. And these cells here, these black cells, are called our endothelial cells. And it makes up, the combined of those two is the tunica intima. So I'm going to write that here in black for those cells. The tunica interna or the intima. All right, so you can put one of the two there. Again, so tunica interna or tunica intima. And again, it consists of these simple squamous epithelial cells with the subendothelial layer under it. All right, so this next brown layer right there, that's the internal elastic lamina. So let's write that there. Internal elastic lamina. Okay? And again, it's made up of, you're going to have some collagen, some fibrillin, and other types of proteins that are present within this layer. This is actually, since we're talking about, I'm just going to mention a quick thing. There is two different types of congenital disorders where you don't make fibrillin or you don't make a specific type of collagen, and it can affect the internal elastic lamina. And these two conditions, I'm just going to write them down here briefly for you. It's called Marfan syndrome, and another one is called Ehlers-Danlos syndrome. And these are both connective tissue disorders. Marfan is usually the lack of fibrillin. Ehlers-Danlos syndrome is the lack of a specific type of collagen. But it affects this internal elastic lamina and other types of connective tissues and can obviously increase the risk of aortic dissections and then aneurysms. So I just thought I'd mention that real quick. All right, so that's the internal elastic lamina. That's the second layer that we want to talk about. Next one is we have a whole bunch of smooth muscle cells all the way around here. Really thick smooth muscle layer right here. This thick smooth muscular layer is kind of actually, but the main three layers is this one out here, and then this middle one. So it kind of gives it the name, the media, or the one in between, right? So I'm going to write it here in red. This thick smooth muscle layer is called the Tunica Media. And the Tunica Media, again, is made up of smooth muscle. And that's really important because obviously it's involuntary. And so we need our sympathetic nervous system to come in and actually innervate this Tunica Media. So I wanted to write that there. This is an important note to have here, is that this is under sympathetic innervation. In other words, it has vasomotor tone. That's extremely important because without the lack of that motor tone, our blood vessels would dilate and we would have neurogenic shock. So the Tunica Media is under sympathetic innervation, which means the sympathetic nerves, it has receptors, adrenergic receptors, present on it. So again, that's our Tunica Media, and it's important for contracting. So it's important for what's called vasoconstriction, constricting the blood vessel and increasing pressure, or dilating the blood vessel and actually decreasing the blood pressure. All right, next one. Again, we have another brown squiggly layer. That brown squiggly layer, it's not the internal now, it's the external elastic lamina. So that last one is called, that second to the last one is called external elastic lamina. And again, it's composed of certain types of proteins, proteoglycans, again, collagen, fibrillin, and again, it's a very important type of structure that allows, basically the purpose of the elastic lamina is to allow for the blood vessel to stretch and to be able to recoil. Without that ability, it's not going to be as kind of elastic and And it can obviously lead to rigidity or maybe some thickening and hardening to be able to expand those arteries if you don't have these elastic laminas. All right, so they're very important for being able to be stretchable, distensible or compliant, and then also recoil. Last one that I wanted to talk about here is this green layer. This green layer is actually going to be called the tunica externa or the tunica adventitia. I prefer tunica externa because it's easier to spell, so we'll put tunica externa. But again, there is another name for it, and you can call it the tunica adventitia also. Now, the tunica externa is made up of a specific type of connective tissue, and I'm going to write that down here. It's dense, irregular, I'm just going to put IR, irregular connective tissue. So it's made up of dense, irregular connective tissue. So it has that gnarled arrangement. The important thing about the tunica externa is it's not supravascular, so it needs a little bit of blood vessels to provide some blood supply to it and maybe to the deeper parts of the tunica media. So if you look here, I drew these small little tiny system of blood vessels right there. So these tiny little system of blood vessels right here are really important, and these are called the vasa vasorum. So they're a tiny system of blood vessels that nourish the tunica externa. Vasa vasorum means vessels on vessels. So it provides nourishment to the tunica externa and some of these actual outer layers of the tunica media so that it gets some oxygen supply. This is actually important because in a disease of tertiary syphilis actually destroys these blood vessels. And if it destroys these blood vessels, it leads to the decreased blood supply to the tunica externa or tunica adventitia, and then maybe damage to the tunica media. And then it can actually lead to thickening of the blood vessel, and then actually weakening of the blood vessel and increased chances of aortic aneurysm. All right, so this could actually be, this is affected by tertiary syphilis, which is caused by a spirochete, also known as Treponema pallidum. All right, so it can actually damage these vasa vasorum here, and it can cause decreased oxygen supply to the tunica externa. And again, that could weaken the walls of the blood vessel and then lead to dissections and aneurysms and emergency situations, right? All right, so everything we went over here and all of these different tunics are exactly the same in this vessel and that vessel. So again, you remember this is the artery, right? And this one over here is the vein. So now what's the difference then? What's the difference? We already know that this is going to have all the same layers. The only difference is in the thickness of those layers. So in a vein, it's still going to have a tunica interna consisting of the subendothelial layer and the endothelial lining. That's still there. The big difference is, and I could have shown it, I could have not have shown it, but the internal elastic lamina is not very abundant inside of the veins because the veins aren't high pressure systems. Veins, if you remember our video on blood vessel characteristics, we said that the pressure of veins is like 5 to 10 millimeters of mercury. So they don't really have to be distensible and stretch. So, but they do have a tiny little bit of that internal and external elastic lamina, but in some veins it might not have any of it. So again, very, very little, if any, internal and external elastic lamina in veins. But very, very little, again, it can have some, usually in the larger vessels. And then if you look here, it has a tunica media, just like the artery does, but what's the difference? The tunica media for a vein is a lot thinner, because if you remember, and I'll say it again, veins are not meant to be very, very high power, high pressure systems and contract to expel blood. They're meant to be capacitance vessels, to be reservoirs, to hold 70% of our blood volume. So it's not going to have a very thick tunica media. It does have sympathetic innervation, so it can contract, but not very much. The most important one right here is the tunica externa. The tunica externa is extremely, extremely thick in a vein, extremely thick. So it has a very, very thick tunica externa in a vein. And on top of that, if you notice the lumens here, the artery lumen versus the vein lumen, when you look at these histologically, one of the things to identify is that a vein, actually when you look at it on a histological or you look at it on a microscope, it's actually going to have a collapsed lumen. So that's another thing. When you're looking on the slides, so you put the slide under, you look in the microscope, you notice that a vein has a collapsed lumen also. So that's really important also. That covers the histology of arteries and veins. All right, guys, let's move over here for a quick recap. All right, so if we look over here again, remember artery and veins, they both have a tunica interna or a tunica intima. It's an endothelial lining with the subendothelial layer. They both have an internal and external elastic lamina. It's just going to be more prominent and more abundant in the artery than it is in the veins. Some veins are not going to have it, the larger veins will, right? And then the tunica media is the smooth muscle layer, and that's obviously going to be it's going to be thicker in the artery than it is in the vein. And then the tunica externa, which is the dense fibrous irregular connective tissue, is more abundant and thicker within inside of the vein than it is in the artery. And again, they both have innervation by the vasa vasorum. Now let's come over here and let's take a look at 3 different types of capillaries. So if we look over here, we have 3 different types of capillaries. Each one of these has some type of morphological difference, and that's what helps us to distinguish them. So if we look here, I'm going to kind of give you the name of each one of these capillaries, and then we're going to discuss each one in detail and the differences within them and where you can find them. So first one here, I'm going to write up here. This is going to be called a sinusoidal capillary. All right, the one beneath it here, which is kind of going to be a giveaway when we see these pericytes here, this is going to be called continuous capillary. So this is called continuous capillary. That's the continuous capillary. All right. The last one here is going to be called a fenestrated capillary. So this is called a fenestrated capillary. Okay, so now that we have all of these capillaries, let's go ahead and discuss each one, why they're specifically given these names, and then on top of that, let's go ahead and talk about where you can find these guys. All right, so first one, sinusoidal capillary. What really makes this one different? Well, if you notice here, we got our endothelial cells that we talked about in the artery and the vein, right? So these are endothelial cells. You're going to notice that they have huge, huge gaps or clefts in between the cells. So these huge gaps right here are called intercellular clefts. So again, what do they have that really makes them distinguishable? One big thing is intercellular clefts, and they are huge, okay? So large ones, we'll put large, okay? So that's one big thing. They're so big that you know that you have white blood cells and red blood cells, specifically red blood cells. Red blood cells can literally leak out of this. Okay, so red blood cells can literally leak out of this blood vessel. Okay, so that's one thing. So if we look here, we can have red blood cells, and these red blood cells can leak out of the capillaries. You can also have plasma. Plasma can also leak out of these capillaries. Okay, so and we can even have large molecular weight proteins. So let's draw here in a molecular weight, a large molecular protein here, like albumin. This can even leak out too. Okay, so these are very, very permeable capillaries, and kind of what makes them specific is these extremely large intercellular clefts. Okay, that can allow for blood cells to leak out, it can allow for plasma solutes to leak out, and it can even allow for large molecular weight proteins to leak out. So where you're going to find this is where you want a lot of cells to be able to move in and out. So this is going to be more commonly found in your bone marrow. Okay, specifically the red bone marrow. I'll write that there at the end. Specifically red. The spleen is a really good one too. Really important one to remember this one. I'm going to exclamation point that one. Spleen is extremely important for this one. And then the last one can be the liver. Okay? All right, so these are going to be the common locations of where you would find these capillaries. Again, you can find them within the red bone marrow, you can find them in the spleen, and you can find them in the liver. And again, what really makes them important? Large intercellular clefts. One last note, these are the most permeable. Most permeable capillaries. Okay, so they're the most leaky, if you will. That's one important thing I want to mention right there. Continuous capillaries, if you look here, they don't really have very large intercellular clefts, kind of small intercellular clefts. All right, and in certain places, like in the blood-brain barrier, they don't even have intercellular clefts because in the blood-brain barrier, the endothelial cells are joined together by tight junctions. But we're going to mention here that there is small intercellular clefts, and we'll put an exception in the blood-brain barrier. So let's write that down here. So let's put here, and this one, let's say, has small intercellular clefts, the smallest clefts, right? And then another thing, let's put a little note here. There's actually going to be tight junctions between these endothelial cells, between endothelial lining and I'm going to put here BBB, blood-brain barrier. This is important, so I want to mention continuous capillaries are found in the blood-brain barrier, but the only difference is they don't have small intercellular clefts. They have tight junctions that link those actual endothelial cells together. That's important, okay? Another thing that they have that none of these other ones have is these really cool cells here, and I'm going to just write them next to them. It's actually called pericytes. And pericytes are believed to control endothelial cell growth. They're believed to be able to cause maybe some vasogenic ability, so vasoconstriction. And they've also been found to act as like phagocytes for things leaking out of the cells. So for this one, small solutes and plasma can leak out through this. And also another cool thing, and we'll talk about this when we get into cellular physiology, is that these guys have the ability to take in little like solutes. Let me actually kind of show you what happens here. I'm going to take one endothelial cell here. That endothelial cell can literally form a little vesicle, an endocytic vesicle, and it can actually take in small molecular weight proteins, and it can actually allow for it to cross to what's called transcytosis throughout the other side here and then leak those things out. This is called penocytosis. It's called penocytosis. So continuous capillaries can actually do that. They have the ability to do what's called penocytosis and just passive diffusion. Okay, that's important. Where would you find continuous capillaries? You would find continuous capillaries where it's got to be very, very controlled. So the main areas you would find this one is actually going to be present within the muscles, the skin. We already said it, but I'll mention again, blood, brain, barrier, and even the lungs. So these are some main areas that you would find the continuous capillaries. Again, what is important about these guys? Small intercellular clefts, but they have tight junctions between the endothelial cells in the blood-brain barrier. They have pericytes, and they have the ability to do what's called penocytosis. And they can also allow for passive transport between the clefts. All right? Let's go into the last one now. So this last one is called fenestrated capillaries. What makes these guys so crazy important or specific? If you look here, they still have intercellular clefts, small intercellular clefts. All right, a little bit bigger than the, I'm going to put here medium-sized intercellular clefts. So let's write that down there. Let's put medium intercellular clefts. Okay, that's one big thing, medium intercellular clefts. And they're also going to have these little, so you see the endothelial cells, there's a little pore between them. There's a tiny little pore running right between them. This little pore, and I'm going to draw it here in brown here, I'm going to imagine like it's a little tube there. So imagine that's like a little tube. These little tubes right here running between those endothelial cells are called fenestration pores. Okay, so they're called fenestration pores. Let's write that down. These are called fenestration pores. And this allows for actually some decent sized molecules to be able to leave the actual plasma. So it can allow for some larger molecular weight proteins, some solutes, some plasma to leak out, but no blood cells. Okay, and again, you had these intercellular clefts, which will allow for passive diffusion of solutes and plasma and stuff like that. And it can also do what this guy can do. So he can also do penocytosis. So he also has cells that can actually do penocytosis. Okay, so that's important. Now, where would you find this? So you want to find this in areas where there's a good exchange occurring. So this is actually going to be more common in the kidneys. So common in the kidneys, and it's very common in glands. Okay, it's common in the glands. And there's two types of glands that we could really describe them. We could say exocrine, which means that they have ducts, endocrine, which means that it is having no ducts. So exocrine is duct glands, and endocrine are going to be ductless glands. And these are some areas that you would find that, the kidneys, the glands, and stuff like that, okay? So, real quick recap of everything again. Let's start here at the top. Sinusoidal capillary, largest intercellular clefts, okay? Most permeable. You're going to find them in the red bone marrow, the spleen, the liver, right? And they can allow for blood cells, plasma, and large molecular weight proteins to leave. Continuous capillary, it's the least permeable. Let's write that down. Let's write that here, right here. Let's put this as the least permeable. That's extremely important. So least permeable here. And again, what makes him different, he's going to have tiny little intercellular clefts, but in the blood-brain barrier, he doesn't have those clefts. He has tight junctions. And again, it has the ability to do penocytosis, okay, through the alveoli. And on top of that, it has pericytes that can control what leaves the actual blood tissue. It also controls endothelial cell growth, and it can control vasoconstriction. And you'd find these within the muscles, the skin, the blood-brain barrier, and even in the lungs. And the last one is these fenestrated capillaries. They have medium-sized intercellular clefts. They have these little fenestration pores between the endothelial cells, and it can allow for plasma, solutes, and then through these fenestration pores, it can allow for some larger molecules to leak out through that also. And it also has the capability of doing penocytosis through those alveoli again. And where would you find this one? You'd find it in the kidneys specifically. That's the best one, and then even glands like exocrine glands and endocrine glands. All right, guys, so that pretty much gives us everything we need to know about capillaries and layers of our blood vessels.